Hello, welcome to our presentation about eggshell-derived nanoparticles for cell imaging application. I'm Colin Lee. So, in this journal that we're citing, they discuss the usage of hydroxyapatite nanoparticles. I'm going to show that to HEP for the rest of the presentation. So, HEP nanoparticles are often used for cell imaging within the body. That's because of their luminescence and their biocompatibility. My friends Usman and Xavier go into more on that later. But what's important is that these HAP nanoparticles are formed using eggshells. Eggshells are ideal because of their low cost and also the fact that they not only contain magnesium, which is kind of the main ingredient for hydroxyapatite, but they also have magnesium. Magnesium makes it even better for use in biological environments without being too invasive. It's more like a natural kind of metal. Now, these nanoparticles take a rod-like shape. They have lengths of about 50 to 53 nanometers and widths of about 18 nanometers. Cell walls tend to be semi-permeable and only let particles of specific sizes in. So this nanoparticle is the perfect size to enter the cell and give us the data we need. The images to the right show transmission electron microscopy images of the nanoparticles I'm talking about that have been derived from eggshells. These nanoparticles go inside and then they return that info.
Hi, my name is Barrett Carter, and I'll be discussing the application of nanoparticles. So the problem at hand is that we're in need of a safe and stable material that can be used to observe or track cells inside a currently living organism. Modern techniques operate by using materials that can emit light to be able to locate cells. However, there are some drawbacks to this method. They're such as they're expensive, the dyes that they use lose brightness quickly, and some materials use heavy metals which are unsafe for medical use because of cell damage. So the researchers in our journal that we are discussing respond to this problem by developing a solution that's both cost-effective and safe for medical use. They're able to accomplish this by using a nanoparticle derived from eggshells and europium. This nanoparticle is applied in cases where they need to view a cell or tissue in a safe and cost-effective method. Let's discuss the application of these nanoparticles in the real world. So these nanoparticles are small enough to enter a cell, and due to its europium ions, it emits a luminescent glow, which allows researchers to view and observe the cell. This is important to researchers and society because cell imaging is a vital tool which allows for a deeper understanding about how cells grow, divide, respond to drugs and diseases, and it even allows for detection of tumors. To be able to do this safely and sustainably allows for less developed societies to have access to important technology.
Hey, my name is Usman Abrar, and I'm going to discuss what these nanoparticles do exactly. So, the nanoparticle is europium doped hydroxyapatite, and scientists don't utilize the thermal and magnetic properties of this nanoparticle, and instead use the luminescence property. So, when you add europium to hydroxyapatite, it changes the electrical property, and it allows it to absorb high energy light. So essentially, what happens is in this nanoparticle, when a specific wavelength is exposed, the europium ion in it will get excited to a higher energy level, and then it will drop down to produce a color that can be seen by scientists. So specifically, if we use 382 nanometers wavelength, the europium ion in the nanoparticle will get excited, and it drops back to balance out the charge. And what happens is that it emits a wavelength of about 614 nanometers, which is the color red. Now, this glowing effect from the part nanoparticle allows scientists to study movements seeing how the color moves within the cell. As Colin was saying, the nanoparticle has a specific permeability, meaning that the nanoparticle can fit perfectly inside the cell without causing any harm or damage. And that allows scientists to inject a certain amount of these and see how movement and how the color moves, and that can allow them to see any abnormalities, such as tumors. The image on the right, in the bottom right, shows how these nanoparticles, HAP doped with europium, is uptaken by other cells, in this case PC12. In the top two images, it has a control variable. Nothing was injected. In the bottom two, HAP was injected. Under the bright field, you can see these HEP nanoparticles clearly. However, when exposed under the blue light filter, there is no HEP being shown. However, whenever there is HEP injected, you can see it shown, which shows how cell imaging can be used by scientists when HEP nanoparticles are used.
Hi, my name is Xavier, and I'll be discussing how does this improve upon the current technology for this application. So to begin, why do we use these nanoparticles for cell imaging? Well, they're small and efficient. They're able to easily enter the cells because they're 30 to 70 nanometers in length and are produce clear imaging. They're also safe and biocompatible. They're made from hydroxyapatite, which is the same material as in our bones. So in total, they're non-toxic to cells and blood. They're also bright and stable. Europium doping gives strong, lasting light, and europium doping is just basically increasing the amount of europium, and it's different from dyes because dyes produce fading. They're also eco-friendly and cheap. This experiment uses waste eggshells, which are renewable and a low-cost source of calcium. And lastly, they're energy efficient. This experiment also uses a hydrothermal process, which uses mild conditions and fewer chemicals. So you might be wondering what problems exist with traditional methods. Well, to begin, MRI and radioactive imaging, well, they're expensive, complex, and may pose health risk. Organic dyes. As I mentioned in my first bulletin point, they fade too quickly. They're also unstable, and in total, they can just damage cells. So the important thing to take from here is that these methods, they do work, but they're either unsafe, expensive, or inefficient, which is why we're always looking for more ways to combat this. So then how do nanoparticles improve this? Well, to begin, they're safer for humans and the environment. Nanoparticles are made of course, hydroxyapatite, which is the material found in bones and teeth, and again, they're non-toxic. They're also a lower cost and energy use. The hydrothermal synthesis method uses water and moderate heat with no energy heavy chemicals, which means they're lower cost and energy use, which makes them efficient. They're also sustainable. This process recycles waste instead of relying on mined or synthetic materials. So basically this experiment is using eggshells, which is basically it's a waste that you would otherwise just throw away. And lastly, they produce clearer and long-lasting images. Europium ions make the particles glow brightly under visible light, creating red emissions with no fade. This is basically what scientists look for. References. Our references came from the Web of Science article. Nanoparticles make life better. Thank you for watching.